One of the most important activities of DNA is that it has to be able to copy itself so that it can spread itself around the world. Every frog, every tree, every flower has to have the DNA in it to make its body. So the question is, if this DNA molecule is a double-stranded double helix, then how does it make more copies of itself? So what we call this process is DNA replication. And the Watson and Crick model of DNA provides an easy model for how DNA replicates. All research that's gone into this seems to verify the predictions of how the model worked. So evidence shows that the DNA molecule unzips using an enzyme called helicase, and that separates the DNA in small areas into two strands. This blue triangle here is helicase, and what it's doing is it's sliding down and it's popping the bonds between adenine and thymine and guanine and cytosine. And what that does is it opens up these single-stranded sections of DNA. And then what happens is special enzymes move in. These enzymes, there's actually a number of them, but the one I'm going to stress is DNA polymerase. The DNA polymerase actually then adds the correct nucleotides into position based on what it finds sticking out on these single-stranded pieces of DNA. So if it finds an adenine here, it'll put a thymine on it. If it finds a thymine here, it'll put an adenine on it. If it finds a guanine here, it'll put a cytosine on it. And if it finds a cytosine, it'll put a guanine on it. So the point is that the DNA molecule is replicated by unzipping it and then rebuilding the parts that are missing on the unzipped parts of the DNA. Now the thing is, the DNA molecule is double-stranded and the two sides are anti-parallel. And as it turns out, the DNA polymerase is, and that's what this big enzyme is showing right here, the DNA polymerase is not actually able to go in any direction. It can only slide in the 5' direction on the existing DNA molecule it's working. This opens a peculiar problem during DNA replication. So when helicase opens up the DNA, one side will replicate smoothly and the other side will replicate in segments. Okay, so when the DNA is unzipped, this is called a fork. These two sections sticking out. So when the DNA is unzipped, the fork has a leading strand and a lagging strand. The leading strand is faster at replicating than the lagging strand. And you'll see why in just a minute, why one side's leading and one side's lagging. Enzymes called DNA polymerase move down the strands and they put the new DNA nucleotides in position, as I was talking about before. DNA will only replicate in sections on the lagging strand because it has to move away from helicase and replicate towards the 5' end. The lagging side requires all kinds of enzymes: polymerase 3, polymerase 1, primase, and ligase. The leading side doesn't require all those enzymes. The little fragments that are created on the lagging side are called Okazaki fragments, named after Reiji Okazaki that discovered them. This kind of replication is called semi-conservative replication because when a piece of DNA is replicated, each new piece, so if one piece of DNA is replicated into two pieces that are copies of each other, the two new pieces are half new and half old. That's because the original DNA unzipped and those old strands were then copied by DNA polymerase, making new half strands to match them. So here's another animation that kind of describes how this works, and I'll go to the controlling one. The little key here is helicase. So helicase is going to move down the line. Here's the fork now. This is one side of the fork. This is the other side of the fork. This is going to be the leading side of the two forks, and this is the lagging side. The reason is the phosphates on the top are facing away. The phosphates on the bottom are facing towards. Now, I made this animation very simple, so I should have started the replication immediately, and I didn't. But on the bottom, the DNA replication is going to proceed from left to right. And on the top, it's going to proceed from right to left. And here's how it works. So on the bottom, DNA polymerase is going to slide down these nucleotides in the 3' to 5' direction. Remember, the 5' refers to the direction of the carbons on the deoxyribose. You can always tell which side is 5' for where the phosphates are sticking out. So as the DNA polymerase comes down, it's a little paintbrush here. It finds a T, so it pastes an A in. Here it finds an A, so it's going to paste a T in. Then it finds a C, so it's going to paste a G in. Then it finds a T, so it's going to paste an A in. It'll find another T, so it'll paste an A in. It'll find a C, it'll paste a G in. It'll keep just going down the line. Meanwhile, notice the helicase went to the right, okay? And if this chromosome was true size, it'd still be going. The DNA polymerase on the bottom is following helicase to the right. But the DNA polymerase on the top is going backwards. It's going away from the helicase, and that's what creates problems as the replication proceeds. So you'll notice that the polymerases, though, faithfully put in the new nucleotides. Let's see if I've got a better animation here. This is a better way to look at this. When DNA is replicated, usually helicase starts right in the middle somewhere of the DNA, and it literally unzips it from the middle. This is called a bubble. It opens a bubble. So this is all single-stranded DNA up here, and this is all single-stranded DNA down here. Right where the helicases start. So a helicase will start here, and that's helicase, and it's going to zip down to the left, and it's going to unzip that DNA that way. And another helicase is going to start here, and it's going to unzip, and it's going to slide down to the right. Right where the helicases started is called the origin. Now, since this is the 5' end on the top strand, DNA polymerase can just slide down and follow the helicase on the top strand. But on the bottom strand, the DNA, as the DNA is exposed, the DNA polymerase will replicate away, because see on the bottom, 5' is over here. That's the phosphate sticking out. So on the bottom strand, DNA polymerase will replicate on the bottom strand here. It'll just replicate smoothly. It'll just follow the helicase on the left side, moving down the line. But this little section right here, this is the lagging section. This is the blue parts of the leading sections. Leading, leading, lagging, lagging. The lagging section is replicated in little bursts. As this DNA is exposed, the DNA polymerase will replicate down until it hits DNA that's already been replicated. There may be 6 different DNA replication enzymes flying around in here replicating this bubble. When this DNA polymerase reaches the blue part that's already been replicated, it'll detach. Then another DNA replicase will attach here, and it'll slide down. When it hits this little section that's already been replicated, it'll detach. Ligase, a special enzyme, is needed to bond together these little fragments. So on the lagging side, DNA is replicated in little fragments, and ligase is needed to bond the little fragments together. On the leading side, no ligase is needed because the replication is smooth and continuous, and all the nucleotides are connected together. But on the lagging side, the nucleotides are not connected together between the fragments, and that's what ligase is needed for. So when chromosomes are replicated, multiple bubbles form with multiple helicases, all kinds of polymerases, and the bubbles grow and they eventually fuse with each other. So what was once, this is a single piece of DNA up here, and they didn't show the nitrogen bases, just for ease of drawing. Now we've got 2 pieces of DNA down here, and what they've got dashed is new nucleotides, and solid is old nucleotides. They call this semi-conservative replication because the replication is producing new DNA molecules that are half new and half old. So that concludes our discussion of DNA replication. It is a complicated process, but it is an absolutely critical process for living things, because without it, there would be no way to make new copies of DNA to repair your body. Every time you cut yourself, your DNA has to replicate in the cells that are around the cut so that new cells have new DNA, well, it's half new and half old, to actually grow and replace the cells that died at the cut. Also, during reproduction, you have to have new Well, it's half new, half old, but you have to have DNA available to pass through sperm and egg to offspring.